So, very good evening, everyone. Is it true that we have only 5 students join us today, or is there any some technical glitch? Sorry, I cannot start the class with 6 students here in the class. I'm sorry. The lectures cannot be continued in this way. Okay. If they want to join, they will join. We can start. I have to make this done. So protein isolation, quantification, and purification. So basically, we're gonna discuss today many techniques in which we can able to identify a protein and can isolate them, and then further, how much protein we have isolated. For the same, we have to do the quantification. Now, after the quantification is done, then comes the purification. That you want to have a pure protein out of your sample. So all these process goes through. So first we will deal with the, in order to isolate your protein, homogenization, fractionation, centrifugation. Then we do the quantitation. Then afterwards for purification, we have many techniques in the chromatography and gel electrophoresis. So all in all, this is the content of today's lecture. So let's discuss. No, it's 15. Where is that? So Archer John Porter Martin. So in 1952, they founded the National Institute for Medical Research, United Kingdom, 1910 to 2002, for the invention of partition chromatography. Then Richard Lawrence Willington in 1952, they also worked in the partition chromatography. Then later on, earlier on, in 1948, Arne Tislas. So he worked on the electrophoresis and adsorption analysis, especially for his discoveries concerning the complex nature of serum proteins. Then comes your in mass spectrometry and sedimentation, John B. F. N., Koichi Tanaka, and Theodor Swedberg. So John B. F. and the development of methods for identification and structural analysis of biological macromolecules. Then Koichi Tanaka in 2002 for the development of soft resorption ionization methods for mass spectrometric analysis. Then Theodor Svedberg in 1926, they worked upon the Uppsala University and worked on the dispersed system. Albert Claude, Christian Dubé, and George Claude. So they together they got the Nobel Prize winner in different years in organelle fractionations to the inner organs, inner organelle of our cell, to fractionate them, to differentiate them, to get them separately. So that was done at that time. Then this our famous professor Frederick Sanger, famous researcher, scientist. So he got 2 times Nobel Prize winner in his life, one in 1958 and one in 1980. The one in the 1958 he got the University of Cambridge in 1918. That is for his work on the structure of proteins, especially of that insulin. Then later on for the contribution of the determine the base sequence of the nucleic acids. So he was the one, Sanger sequencing we have discussed in the techniques in molecular biology. So he was the one who have discovered that. Then how to isolate these proteins? So in order to isolation for proteins, first step is to disrupt your cells, to remove the cell membrane. And then we have to do fractionation, like to separate your various fractions of the cells, various different parts like nuclear, mitochondrial, Golgi, microsomal, nuclear fractions into different parts. So we do the fractionation. So initially for the homogenization, you can use your Dow's homogenizer. Sonication we could use osmotic shocks and French press, right? And for the fractionation, we can go ahead with the fractionation precipitation with ammonium sulfate precipitations and differential centrifugation. Yeah. So homogeneous forms we centrifuge at 500 G for 10 minutes and with a supernatant in the top, and the pallet will have the nuclear fractionation. Yeah, and it will go for 10,000 G for 20 minutes. And afterwards, you will have your pallet, the one that you will have in the pallet, it will be your mitochondrial fraction. Once it is done, then 100,000 G. So after the 1 hour, you have a cytosol, which will have your microsomal fractions afterwards. Then once you have done that, you can also differentiate between different samples of your organelle by centrifugation. So there are 2 techniques to separate your different molecules, that is differential centrifugation and rate zonal centrifugation. So in the differential centrifugation, you have sample is poured into the tube. So you have both more dense and less dense particles. Then you centrifuge it. Then according to that, the dense particles will go down as a pellet and the less dense particles will be on the top. So you can decant that liquid in the container and separate the pellet separately. So in the second case, with the rate zonal centrifugation, sample is laid on the top of the gradient. Yeah, so there is sucrose gradient which will have the mixture of your large particles and small particles. Then you centrifuge it, and according to centrifugation, your molecules will separate it with respect to its higher density and lower density. And then we can separate them with increasing mass of particles in the assay. So that's how you can isolate different proteins, according to their density, according to the mass, with the help of first doing the homogenization, then disrupting the cells, then doing this fractionation technique, and then the centrifugation technique. So things that you will get at the end, it will be like you are interested, let's say, within the when I was doing my case for the isolation of cardiomyocytes, so within the heart there are fibroblasts and there are also cardiomyocytes. So both are present within the when you extract the primary cardiomyocytes. So what we have to do is we have to separate between these two. So in order to do so, we took the sucrose gradient in our case also, and after taking the sucrose gradient, we went for after taking so we separate them according to the density. We put your sample. So within this sample, the one the less dense, that is your fibroblast, they will show a layer ring on the top, and at the bottom you will have your ring of what you say, your ring of cardiomyocytes. Yeah. So you can separate them separately. Then you can, like with the help of pipette, you can remove them and use it for your own work later on. So same for the protein isolation and centrifugation at 300,000 G for 5 minutes, at 10,000 G for 10 minutes, and 40,000 G for 30 minutes. So pellets with the large organelles will be the chloroplast and nuclei could be separated, and at 10,000 G for 10 minutes, your mitochondria and inclusion bodies could be separated, and at 40,000 G for 30 minutes, membrane, plasma membrane, tonoplast, vacuolar membrane, microsomes to be separated. And at that 200,000 G for 16 hours, you can separate large protein complexes like ribosome and even more than 500,000 complexes. At 400,000 G for 24 hours, you can separate protein of around 50,000 Dalton size. So this is all possible, and there is also sedimentation velocity technique here. So you have a low density solution and a high density solution. So you put into your centrifuge tube, and there will be density gradient and centrifuge tube there. So layering of sample is done. You put your sample, again centrifugation will happen. So according to the sedimentation coefficient, all your samples will be separated in your gradient solution. So fractions are collected in the whole of the bottom of the tube. So you keep putting the first blue one, then the sky blue one, then the green one, and the light sky blue one, and then the pink one, and so on. So this is, yeah. Then comes your protein quantitation. So once you have done your fractionation, yeah, your homogenization is done, you have isolated your protein. Now it's time to see how much protein that you have. So basically in the market there are two methods that we do in the lab. That is Lowry method and Bradford method. So the Lowry method, initially it's meant to done the protein determination, which tend to calculate from 10 microgram to 1000 microgram per ml. Whereas the Bradford protein determination, the technique is simpler, faster than the Lowry method, and is subject to less interference. Yeah. So Bradford, whereas the Commercy Blue G250 dye appears to be bind most readily to the organyl and lysyl residues of the proteins. Then they are very specific to a specific assay, and they find quite Bradford give us very nice details at the end. So now let's see if this video will play or not. Oh yes, it's playing. That's good. How Bradford assay is done in the lab.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
Okay. Was the very simple and one of the widely used method in the lab. Whenever you will be joining a lab, oh, today is 30th June. Just a second, student. Good, 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 good. Um, yeah, so this technique is basically if you're having, a, a disease sample and a normal sample, and you want to do a lot of, analysis, protein analysis on them. But in order to do so, first you have to make sure that all the protein in your sample, they are on equal amount. That in your disease state and in the normal state, both proteins are of equal amount and there is no differences in them. In order to do so, your Bradford assay helps you. So you want all your samples to be 5 microgram per ml to be present in every sample. So that's why this Bradford assay is basically helps you to make a strong foundation for your experiments. And somehow, if you're doing with the large proteins, then it will be not so good method. But still, in the lab, day-to-day life, it's being highly used in the lab with the Comacy G250 dye and working pretty good. Then protein quantitation. So this is ultra sensitive technique. So in this biotechnique, development and characterization of nano orange protein quantitation assay is done. So what you have to do is you just add dye to the sample, then heat for 10 minutes and cool to the room temperature, and you can voila, read the fluorescence. This is also one of the latest technique in 2003, has been identified by Jones. Pretty good actually, pretty much good. So this is how you get the results. On the y-axis is the fluorescence, on x-axis is your protein with a concentration of microgram per ml, 2, 4, 6, 8 and 10. And then with the increase in your fluorescence, your protein concentration will be increasing. So once you have identified one fluorescence, and based on that, you can predict the concentration of your protein itself. That's how it's work. And now comes your purification part of a protein, that how we can purify your protein. So we have understood so far the basics, the basics behind how we uh, like isolate the protein, and then we do the quantitation, and then comes the third step, that is purification. This if sometimes if somebody asks you that enzyme purification, in the same case you can tell all the technique of your protein purification, because enzyme is actually a protein. So both will work for both techniques will be the same techniques will be work for. So dialysis, it's a technique to remove your small molecules from the protein solution or to change the buffer composition of the protein solution. And dialysis tubing or dab is made of cellulose and has different cutoff range. So here we have a dialysis bag in which we are having a concentrated solution with the bigger and small molecules. And outside is your buffer solution. Yeah, so at the start of dialysis, uh, all the things will be inside, but after the equilibrium has been achieved, you will see everything to be outside present. Your small molecules are all outside, and the big molecules remain in the inside part. So that's how the protein purification is taken care. So let's see how this is done practically in the lab, the dialysis of a protein. Oops, oopsie. That was the basics behind your dialysis. Very simple, not so difficult technique. Now comes your chromatography, which is divided into different parts, and one of the interesting technique. So this is basically, as we know, in the liquid chromatography, in which you have a chromatographic methods. You have a mobile phase, which is liquid, and a stationary phase could be a liquid or solid. So let's take the first example of your gel filtration chromatography. So in this, you're having three different, like you're having this three different components of solution you want to get separated, right? So here we having these big, these are carbohydrate polymer beads, these big ones, yeah. These are carbohydrate polymer beads, and the small ones are these are small molecules which enter the aqueous spaces within the beads. So they will remain inside the beads, whereas the green one they will tend to go outside, and the yellow ones also tend to flow down. So you can separate them. So first your yellow will be comes down fast, then comes your green, then comes your red. So you can separate them with this, and in this part you will have your molecular exclusion gel, so which will tend to make your protein separated. So slowly they start to separate from each other, and at the end you have your three different proteins getting separated from each other. So here we have a large and small proteins, and these are carbohydrate polymer beads. So the yellow ones tends to go down, and in this carbohydrate polymer beads, your green ones tend to join in, and then later on in the collection fraction we tend to get our samples. So here this is separation by size. This is the Cephadx G75, this is Cephadx G10. This is just telling the sedimentation power of these gels. Yeah, so let's see how we do this practically. Gel filtration Cephadx G50. It's a very interesting method. It's a time consuming. You need a lot of patience to do so. It's not in a single day it could be done. Yeah, in a single day it could be done, but you get a really good results afterwards. So that's the best thing.
We're starting with gel filtration medium. This is Cephadx G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and have made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap, so I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
Okay. Then comes your next, that is your it's lagging. Okay, it will work. So chromatography, ion exchange chromatography technique. So in this, the protein in solution has low ionic strength, which is added to the column, and the protein interacts with the beads. That is your column matrix via the electrostatic interactions. So your resins will be carrying positive charges are called anion exchangers because anions will exchange on this type of column. And resins carrying negative charges are called cation exchangers because cations will be exchanged on this type of column. So you have positively charged proteins bind to the negatively charged beads, and you will have negatively charged proteins which flows through it, right? So all the positive one will attach to these beads, the negative beads, and the negative will flow through it. And this way you have your separated between your two molecules. So here we having like in an ion exchange chromatography, and there are two types now, anion and cation exchange chromatography. The anion exchange chromatography, the matrix carries a net positive charge which interacts with the negatively charged protein strongly, whereas in cation exchange chromatography, matrix is negatively charged and it interacts with the positively charged proteins. So it's a vice versa of your both things. Then comes your sulfonate, quaternary amine, diethylamino, ethyl, and hydroxyapatite. So they have different exchange types, structures, and so on. It seems today our data that what I'm speaking here is it's lagging a lot. So the videos might not play, right? So there's a problem with the videos playing. So in that case, so it will not make sense here to turn this video on because after I speak this video will be very slow, and the learning that you will be doing here, it will be not good. It happens sometimes because in earlier days our videos were working pretty much good, there was no problem. But so what can I do is I can share these videos over the Telegram group, yeah, so you can watch them. So there's a Bradford video we saw. Then we saw a dialysis protein. Then if you are not able to see this gel filtration, try to see this in this one, the gel filtration one. Yeah. So next I want to play with the ion exchange chromatography, but it seems it might not work as it should do. So today we will just concern with this video, ion exchange chromatography. So I will try to play this. If it works, that will be pretty good. That will be awesome. And the lectures of today. Yeah.
Ion exchange chromatography is a chromatography separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into anion exchange chromatography and cation exchange chromatography. Anion exchange chromatography. Negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both protein B and C carry a net positive charge. In addition, protein C is more positively charged since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified, concentrated form.
So, students, I think my internet and everything was working fine, and there was no lagging was happening like earlier. And you also got most of the videos and the presentation also. And the affinity chromatography, we do this tomorrow. Yeah, it's working fine. That's good. Not so much of lagging. Perfect, perfect. Yeah, so we are done for today. So thank you so much. So in this way, we will continue with further chromatography techniques like HPLC, affinity chromatography, 2D gel electrophoresis. Yeah, so these mainly 3 or 4 techniques are left. Then we are done with the protein part also. So not to hurry much today. Tomorrow we will see us again and will continue further. So till that time, take care. Wish you all nice evening. Bye. Good night.